Hello dear students. In this chapter so far we have already studied about the cell cycle followed by the process of mitosis. Today we will take up the last topic of the chapter, which is meiosis. I hope you have understood the process of mitosis, because you can understand meiosis only when you have studied mitosis. So let's recapitulate very fast what we have already studied. So we have already studied about the cell cycle. We know that a cell, before undergoing a next division, undergoes a cell cycle which involves interphase followed by the division. Like in interphase, again there are three stages. G1, where the cell organelles duplicate. S phase, where the replication of DNA takes place. And G2 phase, where the protein synthesis takes place. And then this is followed by the division, generally the mitosis. And once the mitosis is taking place, it results into two daughter cells, and then those two daughter cells again undergo the cell cycle, that is interphase, and then again the cell division. Okay, and again in mitosis we have studied that it is a very sequential division, including karyokinesis, that is division of the nucleus, followed by cytokinesis, division of the cytoplasm. In karyokinesis, you know, there are four stages: prophase, metaphase, anaphase, and telophase. So we have studied up to this process. Today we will take up meiosis. Before going into the details of this process, let us study few characteristics of the process of meiosis. You can see here, number one, meiosis is called as a reductional division. Why? Well, it is because after a cell undergoes meiosis, the daughter cells which are produced, they are not exactly like their parent. They have the chromosomes half as compared to their parent cell. The chromosome number is reduced to half. That's why this is called as reductional division. Therefore, meiosis takes place only in diploid cell. In mitosis, we studied that mitosis can take place in both the haploid cells as well as diploid cell, whereas meiosis is possible only in diploid cell, and it will result in the formation of haploid cells. Okay, so that's why it is called as reductional division. First characteristic. Second, this is a highly complex process, a very long process. So we study the whole process of meiosis in two parts: meiosis 1 and meiosis 2. We will study the details of the process in the further. Then next, after a cell has undergone meiosis and daughter cells are produced, there will be no further cell cycle. Whatever daughter cells are produced, they are used or they are disintegrated. They will not further undergo the cell cycle. Okay. And next, after the complete meiosis has taken place, meiosis 1 and meiosis 2, the resultant will be 4 daughter cells. But these 4 daughter cells will not be identical to their parent. Also, they will not be even identical to each other. They will show certain variations. So, 4 non-identical daughter cells are produced, and these 4 daughter cells may act as gametes. This is the reason Why? There is variations in the siblings of the same parents. Now let us see what are the significance of meiosis. The first significance is the variations. Because of these variations, because of the meiosis, the offsprings produced are not identical to each other. They are showing certain changes. They are showing variations. This is because of a process called as recombination. Okay, so as a result, the gametes which are produced, they are again the recombinants of their parental and paternal and maternal chromosomes, which I will explain you. And then next significance is that meiosis process is responsible for the stability of chromosomes in sexually reproducing organisms. Just imagine if there would have been no meiosis, no reductional division, the chromosome number would have increased in every generation. Therefore, this meiosis is responsible for maintaining the stability of chromosome numbers in sexually reproducing organisms. So, this is the general characteristics of meiosis. Now, let's study the process of meiosis in detail. Now, let us study the process of meiosis. Let us begin by studying it through a flow chart. Here in this flow chart, I have written different stages of meiosis. Just see, meiosis, as I told you, is a complex process and it is divided into two stages, meiosis 1 and meiosis 2. And in between meiosis 1 and meiosis 2, there is a small resting phase which is called as interkinesis. So many a times a question is asked, what is the difference between interphase and interkinesis? So interkinesis is a small resting phase between meiosis 1 and meiosis 2, whereas interphase is the resting phase between two successive mitoses. Okay. So let us see. In meiosis 1, again you can see there are four stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. Out of these four stages, prophase 1 is the longest phase and there are so many changes taking place in prophase 1, and this prophase 1 is further divided into 5 substages. Let's see what are the names of these substages. First is lactotein, followed by zygotine. Third is pachytene. Fourth is diplotin. And finally, fifth is dikinesis. We will study in detail what all changes take place during these substages. Okay. So once the prophase is completed after dikinesis, then there is metaphase 1, anaphase 1 and telophase 1. After telophase 1, two daughter cells are produced. And these two daughter cells, after a short period of rest, they further enter into meiosis 2. And meiosis 2 is just like mitosis. So, it is again both the daughter cells will undergo prophase 2, metaphase 2, anaphase 2 and telophase 2. And as a result, after telophase 2 and cytokinesis, there will be 4 daughter cells produced. Okay, so after meiosis, one parent cell results into 4 daughter cells. Now let us study the whole process of this meiosis through diagrams. So these are the diagrams of different stages of meiosis 1. Meiosis 1 is a very lengthy process, and out of that, prophase 1 is the longest. And as I've already told you, prophase 1 is divided into 5 substages. Most of the changes take place here only. Now see what's happening. We will begin with this cell which is coming from the interphase. During interphase, you know, a cell has got a nuclear membrane, nucleolus and chromatid. Here we are taking 2n is equal to 4. And this means 2 chromosomes are maternal and 2 chromosomes are paternal. That's why I have used 2 different colors, red and blue. Okay. So now this cell will undergo prophase 1. In prophase 1, first stage is leptotene. See what is the changes taking place in leptotene. These chromatin have converted into chromosomes by condensation and coiling. After leptotene, you can see next stage is zygotene, which is very important because what's happening in zygotene? There is synapsis taking place. What is synapsis? Synapsis is the pairing of homologous chromosomes forming bivalent. Okay? So, there are 2n is equal to 4, 2 maternal chromosomes and 2 paternal chromosomes. So, these chromosomes will form pairs, homologous pairs. Homologous chromosomes are the chromosomes having same genes on them, which means chromosome number 1 from father, chromosome number 1 from the mother. Likewise, okay. So, synapsis takes place. Synapsis means pairing of homologous chromosomes and bivalent formation takes place. This takes place during zygotene. Now in pachytene, now you can see here the crossing over between non-sister chromatids of homologous chromosome takes place. What does it mean? This is one homologous chromosome. This is a pair of homologous chromosomes. So, 2 arms of the same chromosomes or 2 chromatids of the same chromosomes are called sister chromatids. And 2 chromatids of 2 different chromosomes are called non-sister chromatids. So, what's happening? Crossing over between non-sister chromatids of homologous chromosome takes place. This is called crossing over, which takes place during pachytene. Now comes diplotene. During diplotene, after crossing over, there is repulsion between the homologous chromosome takes place. As a result, there is the formation of cross-like structure which is called as chiasma. So, at the point of crossing over, there is a cross-like structure taking place which is called as chiasma formation. So, chiasma formation takes place during diplotene. After diplotene comes the dikinesis, last stage of prophase 1. In this case, the segments, they are exchanged. So, you can say recombinant chromosomes are formed. So, you can see here, there is recombinant. So, a segment is exchanged between the non-sister chromatids of homologous chromosomes. As a result, recombinant chromosomes are formed. So, after prophase 1, the cell will now enter into metaphase 1. In the metaphase 1, what's happening? You can see the spindle fibers are formed and all the chromosomes are lying at the metaphasic plate or equatorial plate. Okay. Now comes the anaphase. Anaphase, again, there is stretching about the cell. Anaphase 1 is actually the phase where reduction begins. In mitosis, we studied that during anaphase, the chromatids separate. And half of the chromosome, those chromatids which become chromosomes, half move to one pole and other half move to other poles. But in this case, there is no separation of the chromatids. In fact, out of the pair of the homologous chromosomes, randomly one chromosome from one pair moves to one pole and other member of the pair moves to opposite pole. So half of the chromosomes will move to one pole and other half will move to other pole randomly. So you can see these two chromosomes are moving to this pole and these two chromosomes are moving to this pole. So as a result, you can see the number of chromosomes have reduced. This is telophase 1. Telophase 1 is just a reversal of prophase. Again the nuclear membrane has reappeared and then the chromosomes elongation taking place. So you can see here, out of the 4 chromosomes which the parent cell had, now the 2 daughter cells which are formed after meiosis 1, they will have only half the number of chromosomes. Out of 4 chromosomes, one cell will have 2, other cell will have other 2. So this is the end of meiosis 1. Then meiosis 1 to meiosis 2, there is a small gap. Only the centrioles are multiplied and then it again enters into another division which is called as meiosis 2. Let's study that. After meiosis 1, let us now study meiosis 2. In meiosis 1, the 2 daughter cells which are produced after telophase 1, they now enter after a short gap into meiosis 2. Meiosis 2 begins with prophase 2. Now in prophase 2 you can see each cell has got half the number of chromosome as compared to the parent cell. Now again it has got nuclear membrane which will disappear, nucleolus which will disappear, the centrioles will start moving to opposite poles. Okay, now you can see here, this is metaphase 2. One daughter cell is this, another daughter cell is this. This will also undergo the metaphase 2. This will also undergo metaphase 2. So in the metaphase 2, all the chromosomes, they will come at the equatorial or the metaphasic plate. You can see here, these are the recombinant chromosomes. Okay, now comes the anaphase 2. Anaphase 2 is just like the anaphase of mitosis. So these chromatids which are present, they will separate into chromosomes and they will move to opposite poles. So you can see half the chromosome will move to another one pole and other half will move to another pole. Like this here also. Now this is telophase 2. Telophase 2 is just like the reversal of prophase 2. So what will happen? The nuclear membrane will reappear, nucleolus will reappear, the spindle fibers will disintegrate. Okay, and you can see from each cell 2 daughter cells are produced, and after cytokinesis from one parent cell 4 daughter cells are produced, and all these 4 daughter cells, they are not identical. They have half the number of chromosomes, but the type of chromosomes in terms of the recombination is different. So from the same parent cell, 4 daughter cells are produced which are different. They are not the carbon copy because they are recombinants, and as a result 4 different types of cells are produced. Okay, and these cells will act as gametes. That's why the gametes, they show variations when they fuse with another gamete of other parent. Okay, so in this way, in meiosis, we can study that this meiosis is a very elaborate type of cell division which involves the formation of 4 haploid cells. So with this, we have completed the chapter. So before winding up, we must recapitulate the differences between mitosis and meiosis. So how are mitosis and meiosis different? Mitosis is an equational division, whereas meiosis is a reductional division. Mitosis can take place in both haploid as well diploid cells, whereas meiosis takes place only in diploid cells. Mitosis results in the formation of two daughter cells, whereas meiosis results in the formation of four daughter cells. Mitosis produces exactly similar daughter cells. The daughter cells are clones of each other as well as clones of their parent cell. Whereas meiosis produces four daughter cells which have half the number of chromosomes, and those four daughter cells are also not clones of each other because of the, you can say, the crossing over between the homologous chromosome. Okay, mitosis is mainly responsible for growth, for healing, also for asexual reproduction, which produces clones. Whereas meiosis is mainly responsible for creating variations among the organisms. It is also responsible for maintaining the stability of chromosomes in sexually reproducing organisms. Okay, so with this we have completed the chapter. The chapter is short, but it is very important. Please practice the diagrams. Each stage is very important. This is very important from NEET point of view also, as well as from exam point of view also. In class 12th, we will be studying the genetics, and genetics is based on lot of these cell division. So if you are clear with cell division, it will be very easy for you to understand genetics concepts in class 12. Okay, thank you.